meta-arterioles are an intermediate vessel that is in between the capillary and the arterioles. They basically provide a bypass that shunts blood from the arteriole directly to the venule. So there are times where we need to bypass the capillary bed, and this is done via the meta-arteriole. So it directly connects the arteriole on one end to the venule on the other end. And there are rings of smooth muscle that are at strategic locations in the meta-arteriole, and they can relax or constrict, and that will regulate the flow to the venule or to the capillary bed. So when the meta-arteriole smooth muscle contracts, that is going to shunt blood into the capillary. So it's going to kind of get opposite from what we've been seeing so far. So constriction of that smooth muscle that's wrapping around the meta-arteriole is shunting blood into the capillary, whereas relaxation of that smooth muscle shunts blood to bypass the capillary and go directly to the venule, which is completely bypassing the capillary bed altogether. Now, this is regulated usually in response to local factors, which we've already talked about, so we're not going to go into that again, but carbon dioxide, et cetera, and that's depending on the state of the local tissue. So if we need to, we can constrict the meta-arteriole, or we can allow flow to go directly to the venules. Before I move from this slide, I do want to point out that it's not quite clear the effect of the nervous system, the sympathetic nervous system, and hormones on this level of regulation. But what we do know is that local factors affects this regulation. But the effect of the neurotransmitters and the hormones is not quite clear. For precapillary sphincters, we know for a fact that only local factors affect the state of precapillary sphincters. And these are basically smooth muscle rings that are lining in between the arteriole and at the start of the capillary bed. So these are also regulating flow into the capillary or to bypass the capillary as well. So they can contract in response to local factors, or they can relax if there's lots of oxygenation and we want to increase flow. So basically when they contract, this constricts the capillary and this drops the amount of blood that's flowing into the capillary bed, and it's shunted away from the capillary and into the venule. If it relaxes, this increases the flow into the capillary and shunts it away from the meta-arteriole. So it's going to get to actually perfuse the tissues through the capillary bed instead of bypassing. So the metabolites that cause relaxation are things we talked about before: carbon dioxide, acid, potassium, etc. Okay. Any questions so far? Any questions? Any questions? Okay, let's start this over. So let's just kind of make sense of all these factors and make some important distinction. So the factors that affect capillary flow: arterioles, meta-arterioles, and then precapillary sphincters. And we're going to speak about these on three different, premises. We'll talk about the location, First, we'll talk about the smooth muscle distribution. Then we'll talk about the direction of flow. And then we'll talk about their regulation. So arterioles have a uniform smooth muscle. Meta-arterioles are just a band, right? Just a band here as well. So just strips of smooth muscle. The direction of flow, arterioles flow to capillary beds. Meta-arterioles flow to where? See if you can remember. To venules, right? So they bypass capillaries to venules. And then precapillary sphincters flow, control flow to capillaries as well. All right. Now the regulation. When arterioles contract versus when they relax. So when arterioles contract, what happens to flow? I'm gonna have you guys kind of help me here. What happens to flow when arterioles contract? Decreases. We decrease flow. Increase resistance or decrease flow. And when they relax, we know we decrease resistance or increase flow. What about when meta-arterioles contract? What happens? When meta-arterioles contract, what happens to flow to capillaries? Yep, so they increase flow to the capillaries. So we increase flow, and when meta-arterioles relax, we decrease flow to capillaries. Okay, remember the meta-arterioles are shunting or bypassing the capillary. And then lastly, the precapillary sphincters, when they contract, what happens to flow? So when the precapillary sphincters contract, right, they're at the start of the capillaries, what happens to flow? Decreases. I think I'm hearing it. Decreases. So this will be decreasing flow. And if the precapillary sphincter relaxes, that will increase flow. Okay? All right, so these are the three factors that regulate flow to capillaries. And because of their different locations, they're going to bring about different effects on the flow to capillaries, okay? Any questions? Any questions on that? All right. Okay, so let's sort of begin looking at the third part of capillaries, which is diffusion. So this is where things are going to get a lot more interesting. We have the movement of fluid across the capillary wall into the intracellular fluid, which is a part of the extracellular fluid compartment, okay? So going way back to ANP1, that very first unit, we talked about those fluid compartments. There was the ICF, then the ECF, and the ECF had two further divisions, the plasma, which is inside the vessel, and the interstitial fluid, which is everything else, right, around the tissues and around the cells. So the free flow of fluid from the capillaries into the interstitial fluid helps to balance out our ECF compartment. So the types of molecules that can easily move across capillary walls are lipophilic molecules. Anyone remember why? Yep, so they can go through that phospholipid bilayer, right? So lipophilic substances can easily cross the membrane. Lipophobic substances need a channel or a carrier, right? Like glucose, it needs to be moved across the membrane with some help. And then we can also facilitate the movement of exchangeable proteins. And I'll talk about what those are as we go on. But via transcytosis, which is a process of endocytosis and exocytosis, we can also move proteins across the membrane. Now, the brain is sort of, again, exempt from this rule. In the brain, things cannot freely move from the vasculature into the brain tissue. What stands in the way? What stands in the way here? So think about astrocytes and tight junctions. So the blood-brain barrier, right? The blood-brain barrier is heavily mediating the flow from capillaries into the brain, and that makes sense. That is to protect the brain from harmful substances like antibodies, pathogens, bacteria,